Hi everyone, in this video we will discuss insulin, insulin structure, synthesis of the insulin, secretion of the insulin and metabolism of the insulin. Insulin is a very important hormone. It is an anti-diabetogenic hormone. It means it prevents us from the diabetes. And we give the insulin in the treatment of diabetes. Okay, so insulin is an important hormone and it is anti-diabetogenic. It is secreted from beta cells of pancreas. So we have pancreas in our abdomen just near to the first part of duodenum, okay, and this is the retroperitoneal structure, means it is behind the peritoneum, and this is secreted from beta cells of pancreas. Then we will discuss the structure of the insulin, how the insulin look like and what type of hormone it is. So it is a peptide hormone. It is a peptide hormone. Peptide hormones means which is made up of amino acids. So this is a building block of a protein, amino acids. So many amino acids makes the peptides, many peptides makes the protein. Okay, so this is a peptide type of hormone. Hormone can be of many type, different structural, you can say that there is a different structure of any hormone. Here it is a peptide hormone which is made up of amino acids. So how it look like? It has two chains of amino acids. Fine, like I am just making these rounded structures. They are amino acids represented as an amino acids. So one is A chain, another is B chain. Fine. So there are two chains, A chains and B chains in the structure of insulin. This is the structure of insulin. And you know that A chain and B chain both are made up of amino acids. So multiple amino acids in a particular sequence make a A chain. Another is the amino acids, multiple amino acids in a sequence make the B chain. And they are attached with the dice by the help of disulfide bond. There are two sulfides, two sulfur, fine. So this is bisulfide linkage or bisulfide bond. There are two sulfur, one sulfur, two sulfur. So that's why it is known as bi. Bi means two. Bisulfur, so bisulfide bond. A chain, B chain and attached with the help of bisulfide bond, it makes the structure of insulin. Okay. Another important thing is a segment of a string of another amino acids which is known as C-peptide. I am just making diagram. You can make in a rounded structure as well. It's a C-peptide. So C-peptide is attached with this A chain and then it makes another molecule which is known as proinsulin. Now this is known as proinsulin. So insulin is made from, like this proinsulin, there is a C-peptide cleavage, C-peptide breakage, and then it makes the insulin. So I can say that insulin doesn't have the C-peptide, okay? But proinsulin structure has C-peptide. Understand? How this look like? See this? This is an enlarged diagram. See. This is the structure of insulin where there is a chain. This is A chain. This one is A chain. This one is B chain. So 2 chain attached with the disulfide bond makes a structure known as insulin. And when this A chain, this is B chain, when with the A chain there is a C chain attached, or C chain, or you can say it is C peptide, then it is known as proinsulin. Before insulin, you can say pro means just before it. Fine. So there is a cleavage of this C peptide, and then it become a insulin and C peptide. Fine. Now, how this is formed, and just see this another diagram also. Now I will enlarge this diagram. Okay. See, this is this structure, how it looks like. There is a A chain which is made up of multiple amino acids, glycine, leucine, valine, glutamate, whatever. There are so many different type of the amino acids, making the A chain, B chain, and attached with the sulfide bond. You don't remember the number of the amino acids where it is attached. Just have to remember that there are disulfide linkage. And if you want to remember this 21 and 30, you can remember this digit as well. This is made up of 21 amino acids, and B chain is made up of 30 amino acids. 21 and 30. This is A chain, this is B chain. The number of amino acids is 1, 2, 3, 4, 5 in that way. So you can remember one is approx 20 and 30. We remember 10, 20, 30 is an easy way. So 20, 30 in that way. It is 21 and then 30. Fine. So these are the amino acids of in the chain A and the chain B. Okay. Now I am just telling you what is the difference between this insulin, proinsulin and pre-proinsulin. One more term is pre-proinsulin as well. Okay. I am enlarging another. This is the image showing the difference between pre-pro and C. Okay. Now see the difference between pre-pro and the main insulin. See, this is the insulin having A chain and B chain. Here it is, they have made it opposite, that A chain below and the B chain is above. There is no difference. They are actually A chain and B chain. Finally, they become folded in the Golgi apparatus. Okay, and they make the insulin structure. So it's okay. Maybe A chain or B chain you can make up or down, whatever you want. So it is A chain, B chain. Just have an idea that the A chain, this peptide C or the C chain should be attached with the A chain only. You have to remember this point. If you are making A chain like this, B chain like this, so attach C peptide here. If you are making A chain down and the B chain here, you have to attach C peptide here only. Fine. So that is important. So this is A chain, B chain, and with the A chain there is a C peptide or C chain is attached. Then it is known as proinsulin. Fine. Another is pre-proinsulin. Pre-proinsulin is with the A, B and C. There is a N-terminal signal sequence. N-terminal signal sequence. What is it? I am just telling you. Okay. Now see what is the difference. See here it is on the N-terminal of B chain there is a signal sequence, or you can say this is also known as signal peptide. And also the name attaches N-terminal signal peptide because this signal peptide is at the N-terminal end. Actually one end is N-terminal where it is NH2. Fine, nitrogen is there. Another is C-terminal. See here, this is the N-terminal, this is a C-terminal. Again in the all chain there is a N-terminal and C-terminal. Fine. So at N-terminal there is a peptide part. You can say a short portion of the amino acids. This is a peptide. any amino acid A1, A2, A3, whatever, there is a sequence. You don't need to remember that sequence of amino acid, but just have an idea that different amino acids in a particular sequence, and they make a small strand that is attached with the C, the end terminal of this chain B, and it make a pre-proinsulin. Fine. Now what is the importance of this? Actually, during synthesis of this insulin, what happened? Initially, this structure is made. Pre-pro-insulin. So, initial molecule of the synthesis of insulin is pre-proinsulin. Pre and pro. You have to add the two words before insulin. Pre-proinsulin. Fine. Then it cleaved and it make the proinsulin. So remove the word pre before this. This is a proinsulin is remaining. Then insulin. Remove the pro word also. So initially you have to remember that how the synthesis occur. Pre-proinsulin, proinsulin, and then the insulin. So first of all you remember these three terms. Now where they are making, where they are synthesized, and how they cleaved or where they are cleaved. Okay. So it is a peptide hormone. You know, peptide is made up of the different amino acid sequence. Any peptide hormone or any protein thing is made up of in a cell machinery. Where is the machinery for the protein synthesis? We know that it is a endoplasmic reticulum. Okay. We know that it is in the cell there is nucleus. Fine. There is endoplasmic reticulum like this. Rough endoplasmic reticulum. So this is the site in the rough endoplasmic reticulum. On the rough endoplasmic reticulum, which is the site for protein synthesis? Ribosome. Okay. So on the rough endoplasmic reticulum, there is ribosomes where the protein synthesis occur. Okay. So similarly here in this protein hormone or a peptide hormone, it is synthesized in the rough endoplasmic reticulum. Okay. So what happened? This pre-proinsulin is synthesized in the ribosome. Then what happened? Out of this N-terminal peptide sequence is removed. Fine. So this N-terminal peptide sequence is removed. See here it is showing again. It is already showing that N-terminal signal sequence is removed from this molecule, and this another molecule is made which is known as proinsulin. Understand? Now what happened? This proinsulin, where it is? It is in the endoplasmic reticulum. Okay? Because the ribosomes is at the endoplasmic reticulum, and in endoplasmic reticulum again some changes occur, and this protein, this C peptide or protein, chain C again is removed out of this and make the insulin. Okay? So here it is removed, the C peptide. But what happened? This C peptide remain along with insulin in the secretory granules. Secretory granules, they are formed at the level of Golgi apparatus. So Golgi apparatus, at the level of Golgi apparatus, there is this secretory granules are made, and the secretory granules are made, and this C peptide is released along with the insulin. This is a whole thing which is a secretory granule. Where the C peptide is there, insulin is there. Okay. So this is very important point, that whenever the insulin is secreted, equimolar amount of the C peptide is also released. Equimolar means the same molar amount. Fine. And how it is important now we will discuss it. So just you have to remember that from pre-proinsulin make the proinsulin, from the proinsulin there is a formation of insulin, pre-pro, pro and insulin. This is made at the ribosome, this is formed at the level of ER, and then Golgi apparatus. At the level of Golgi apparatus, final cleavage and the final folding, this A chain and B chain also fold together and they make a 3D structure of the insulin. Okay, so that this is formed at the level of Golgi apparatus. Fine. Okay, now just see this diagram. See the word secretory granule. This is the word secretory granule. This is actually a secretory granule where the insulin and C-peptide is released together. Okay. So what is the importance of C-peptide? Actually, you can measure the concentration of C-peptide. And you know, whatever the concentration of C-peptide, the insulin is released in that proportion. Okay. Because they are released in an equimolar concentration in the secretory granules. So what you can do? You can do C-peptide measurement. C-peptide measurement. By which technique you can measure? There are multiple techniques to measure the C-peptide or any other molecule. One is the radioimmunoassay. Fine, we will discuss this radioimmunoassay in a separate video. So radioimmunoassay, and there is a measurement of C-peptide by which we know that how much is the endogenous insulin secretion in the body. Fine. Actually, insulin, either it is produced in the body, the endogenous insulin means it is producing inside the body. It is producing inside the body. If a person is having diabetes, I am taking just an example of type 1 diabetes. There are two type of diabetes, type 1 and type 2. In the type 1 diabetes, there is an insulin actual deficiency. So, because there is an actual insulin deficiency, if you measure the C-peptide in these diabetic patients, you will find the decreased concentration of this C-peptide because this endogenous insulin secretion is also lesser there. So, for many patients, we give insulin from outside. There is an exogenous insulin we are giving, or you can say we are giving the insulin injection. We give subcutaneous injection just below the skin, subcutaneous injection we give insulin. So we know how much is the endogenous insulin by measurement of the C-peptide and how much is the insulin which is giving from the outside. So it helps in the you can say to follow the treatment and to know how much our pancreas is working for the insulin production. Okay, so we discuss the structure, we discuss the synthesis and the release. Here the secretion is in the form of secretory granules, and with the insulin equimolar amount of C-peptide is also released. Now when it comes into the circulation, what happens? Fine. So what happened? How it circulate? How there is a circulation of insulin? When the insulin is released, it circulates in the blood and it circulates in the form of unbounded form. Actually, there are two types of form. One is bounded form of hormone. Bounded form is it is attached with the protein and then it moves to the circulation, and whenever where is the required or where is the organ where the action of that particular hormone needed, this hormone release from the protein and then it start acting. Fine. Unbounded form it is not bounded with the protein. So how the protein binding is helpful? Whenever a hormone is bind with the protein, it may act for longer time. So it increases the action of that hormone. So it becomes a little longer acting. But it is because it is an unbounded form, and actually its half-life is very less. Half-life is very less. How much it is? It is approximately 6 minutes. Half-life is very less, it is 6 minutes. So approx in 10 to 15 minutes, the blood concentration of this is very less, so it becomes like you can say metabolize and go in the blood. It remains in a very less concentration. Where it is metabolized? It metabolizes in the liver mainly. Fine. In the liver there is a insulinase enzyme. Insulinase, actually we put the word ase, which is somewhere, which is you can say a splitting type of enzyme. We would use ase, like lipase, okay, so amylase. So all these are the enzymes for the name here. It is insulinase, so which breaks the insulin, that is mainly in the liver. Some amount also found into the kidney, muscles and other tissues, okay. Actually, whatever the, suppose this is a cell, and in this cell there are, or I will make a beautiful one. In a cell there is receptor. This is all receptor. Fine. Receptor is again maybe a different type of molecule, maybe protein, maybe any other chemical. So this is just a receptor. Here we are talking about insulin, so we are talking about insulin receptor. Every hormone, any chemical, any neurotransmitter can have different type of receptors. Okay, so here we are talking about insulin receptor. We will discuss in the next videos in the detail how this receptor look like and how this insulin work on that. Fine. So suppose whatever insulin is put here, once this insulin attach here, it will take some time to act into the cell. So there may be different cellular mechanism how it work on a cell. So once it binds, it may take time, but if it is a free form, it's the unbounded form, it will not remain for longer time. It's a free form, it will not remain for a longer time in the circulation. Fine. So half-life is 10 to 15 minutes. It will remain for the blood, and in the 10 to 15 minutes it's wash out from the blood, you can say. Fine. So this is about the synthesis, the structure, the synthesis, and little bit about the secretion. We will discuss in detail the secretion. What are the factors which helps in secretion? What are the factors which reduces its secretion? In detail in another video. Okay. And then we discuss how this it will go to the circulation in the blood and how it is the metabolized in the body. Thank you so much.